My name is Sivagnanalingam, and I'll be speaking on behalf of Natalie Swavely. Our abstract is on a novel method to evaluate neuroexcitation changes in response to audiovisual urgency triggers using functional near-infrared spectroscopy. We have nothing to disclose. Audiovisual triggers may lead to or exacerbate sensations of urgency in certain individuals with overactive bladder. Functional near-infrared spectroscopy of the brain can be used to quantify neuroexcitation in response to bladder filling. The aim of this preliminary study was to determine whether fNIRS is a feasible tool for non-invasively identifying objective changes in neuroexcitation in response to audiovisual triggers of urinary urgency. For our study, we had participants fill out a survey, and based on their response to the survey, we grouped them into healthy and overactive bladder groups. Accelerated oral hydration study, while brain fNIRS data were collected using an Artenis Bright 24 head cap, as shown on the image on the left. In addition, real-time participant-reported bladder sensation was recorded on a 0 to 100% scale using a tablet-based sensation meter, as shown on the image on the right. Participants watched and listened to a 3-minute video containing scenes and sounds of expected triggers or urinary urgency at 50% bladder sensation. These scenes included images of restrooms, running water, fountains, rain, waterfalls, and swimming. fNIRS oxygenated hemoglobin data were obtained with the receiver located just above the right ear, corresponding to known cortical areas of bladder neuroexcitation. Data from individuals with relatively flat oxygenated hemoglobin during the control period were available for 2 healthy participants and 3 overactive bladder participants. Of the participants, 80% demonstrated an increase in oxygenated hemoglobin during the video. Here, we have an example of oxygenated hemoglobin data for a participant with overactive bladder demonstrating relatively flat oxygenated hemoglobin in the control period, as shown in the figure on the left, and an increase in oxygenated hemoglobin during the fountain and rain portion of the trigger video, as shown in the figure on the right. Also, the red line on the figure on the right corresponds with an increase in sensation reported by the participant. Here is an example of a cortical mesh plot of oxygenated hemoglobin for a participant with overactive bladder. Neuroexcitation in the region of interest, as highlighted by the black rectangle, increased from the start of the control period, as shown in the figure on the left, to the first peak in neuroexcitation during the trigger video, as highlighted by the figure on the right. In conclusion, fNIRS may be feasible for the objective quantification of neuroexcitation changes evoked by audiovisual triggers of urgency. Additional studies are necessary to determine whether non-invasive fNIRS can be used to identify patients with brain-associated overactive bladder. Our study was supported by the following research grants and funds. Thank you.